What's up, guys? Acosta here. Let's go over the formation of the bony skeleton. So there are two main processes that we are going to discuss. One is going to be endochondral ossification, and the other is intramembranous ossification. So what's going on with these two different processes? We are trying to get the formation of bone. What we start off with, that is going to differ. So during the process of endochondral ossification, it says it in the name, chondral, we are referring to cartilage. We are going to start with hyaline cartilage, or what the term of the bone, it's called endochondral bone. During intramembranous ossification, this is a process. The bone is going to develop from a fibrous membrane. And the terminology is a little bit different here. We call this membranous bone. Using hyaline cartilage or using this fibrous membrane, what it's going to allow is some flexibility here. So what it's going to enable, it is able to accommodate mitosis of the embryonic skeleton. And so you can see the process of endochondral ossification. That is what is going to be seen here. So these are some of the steps for endochondral ossification. Before we get into the detailed steps of what's happening during stage 1, 2, 3, 4, and 5, we're going to talk about some of the generalized things. We can also discuss the timeline of when these steps are occurring. What are some of the general things that are going on? One is you can see the formation of what we have, which is the hyaline cartilage. You can see that all the way along here. So this is all of this hyaline cartilage. So the type of bone that this is a long bone. So some of the basic parts of the bone, this is the what's known as the epiphysis, and then this part, that's that shaft in the middle, that is what's known as the diaphysis. If you see this circle here, this is what's known as ossification center. The primary ossification is what's center is formed here. And so we are replacing that cartilage with bone. So it's starting here in the center, and then as you can see, as you go through each of these steps, it's starting to work its way outwards to get the formation. We can see all of this spongy bone here. What we can also see where hyaline cartilage is going to be left. So one is at the epiphyseal plate. So that's one right there. And then it's also found here. You can see this is the articular cartilage. So the timeline in which what's happening here. So during the first step, this is occurring during month 2. So during steps 1 and 2, both of these processes, this is all going to occur during month 2. Once we get to the third stage, so step 3, that is when we get to month 3. Once we get to step 4, so step 4 is going to be occurring between month 4 all the way until month 9, which is birth. So after birth, that's whenever we get the formation of where the hyaline cartilage is only found in these two locations that we discussed. What are some of the cells that are going to be involved here during this process in order to enable it to happen? One is we have these mesenchymal cells. So these mesenchymal stem cells found here within the perichondrium. And these mesenchymal cells, they can differentiate into osteoblasts. So blast means build. So with osteoblasts, we are building bone, and the bone that's going to be secreted, what we know it as, its term osteoid. So that's one way where we can get the formation of osteoblasts. Another formation is to start off with what's known as the osteoprogenitor cells. So osteoprogenitor cells, they can also develop into osteoblasts. If we say osteoclast, what that means is that we are breaking bone down. So why do we need to have osteoblast and osteoclast? So you can see here, this is what's known as the medullary cavity. We're able to get this medullary cavity due to the use of these osteoclasts, which will go ahead and break this bone down. You can also see the formation of this is the periosteal bud. So during the third month here, we're getting that periosteal bud to get the blood supply. Because think about all these different cells that are doing their thing. They have to meet the energy demands, right? They need all the nutrients they can get in order to continue to build some of this bone. So we talked about cell differentiation, which is required for the bone model, meaning when the mesenchymal cells develop into the osteoblast and when osteoprogenitor cells develop into osteoblast as well. Bone degradation, we talked about that, use of those osteoclasts in order to break down some of that bone. Another thing here, we've got bone elongation. So some of those cartilage cells, those chondrocytes, they are going to enlarge or hypertrophy. They'll secrete collagen, the extracellular matrix, and then also we'll talk about how these enlarged chondrocytes are going to secrete calcium. Those ossification centers, what we talked about, right? The primary one here, and then secondary ossification center, that's what forms here. That is where we secrete what's known as osteoid, or that bone tissue, to get the formation of that spongy bone, right? You can see spongy bone found here, spongy bone found here also within, this is the medullary cavity. So what cells differentiate? We've already discussed these different things. The mesenchymal cells, they develop into osteoblast, and so do the osteoprogenitor cells. And what happens with these chondrocytes? They are going to enlarge or hypertrophy, and they're also going to die. So once these guys die, it opens up this cavity here, and then we also have the osteoclasts, which are helping to break it down. Okay, so now let's get into the details. So we've already said that this is the first stage for endochondral ossification. And we said that we get the formation of our primary ossification center. We have these osteoblasts that are going to be secreting that osteoid. But where do osteoblasts come from? So that leads us to specialization into osteoblasts. So these osteoblasts, they're going to be derived from our mesenchymal cells in that first step. And the location of where this primary ossification, it's within the center of the diaphysis. What will happen, we're changing that perichondrium, we're changing that cartilage into our periosteum. Another thing that's going to happen during that first step is that we have these different chondrocytes. These different chondrocytes, they're going to enlarge or hypertrophy. Okay, so that's during that first step, and we've already identified what type of cartilage this is. This is hyaline cartilage, and then over here we get the formation. This is what's known as our bone collar. Okay, so now we'll move on to step 2. What is going on here? So the cartilage calcifies at the center of the diaphysis, but how does it calcify? That is due to the role of our chondrocytes. So our chondrocytes, they're going to secrete this calcium and calcify the cartilage matrix. And so what will happen is this matrix right here, it starts to deteriorate. But why does it start to deteriorate? Well, due to the secretion of that calcium, so it makes it impermeable to the diffusing nutrients. So as a result, those chondrocytes that are found here, they're going to die. So they start to die, and we start to get the formation of our cavity. And so what's going to stabilize this is what we talked about over here, which was that bone collar. So that bone collar is going to be stabilized here, even though we've started to get the formation of this cavity. And so elsewhere, it talks about how the cartilage model is going to elongate. Right, we can see here, if we look at from here to here, right, it's starting to elongate due to the division of these different chondrocytes here. Now we move on to step 3 here, and one of the main things is we get the formation of our periosteal bud. So we've talked about that a little bit earlier, how we get the formation of this periosteal bud for the movement of some of these nutrients. And then you can also see the formation of some of that spongy bone. So that's all the spongy bone found within the diaphysis. All those different chondrocytes, they're going to die. It starts to deteriorate there in that second step, but what other cells are going to be involved to break down some of this? It's not going to be our osteoblast, it's going to be our osteoclast. It will start to erode the cartilage matrix. And then in order for us to get the formation of that spongy bone, what do we need? We need our osteoblasts. And so our osteoblasts, they are going to be derived from our osteoprogenitor cells. And then we get the formation of that spongy bone. And so some of the other things we talked about was the periosteal bud. And we also have the different types of cells here. these different cells, what these cells are going to include is our osteoprogenitor cells, as well as our osteoclasts. Right, that's how we're able to, like what we talked about, the formation of that spongy bone, because once we get the formation of that periosteal blood, these cells are also going to start doing their thing in this part of the bone. Step 4, and we can see the formation of our medullary cavity. So we now have some of that osteoclast activity. They're starting to break that bone down and see how that cartilage, it's being chased in this direction, right? We're replacing that cartilage with the bone in both of these different directions. The diaphysis is elongating, what we talked about here. The growing epiphysis is going to be composed of cartilage only. All of this is just cartilage here. So the next thing to discuss is the secondary ossification centers at the epiphysis. So that's what's going on here. This is our secondary ossification center, and there's differences here with, depending on how the bone is classified, will determine, first off, whether there are secondary ossification centers and where these ossification centers are going to be found. So with long bones, it'll be found on both of the epiphyses. These short bones are going to have a primary ossification center only. For irregular bones, there are several distinct. So we have several of these ossification centers that are going to be found. If we think about an example of an irregular bone, think about the vertebrae. So the last step here, this is step 5. Keep in mind the timeline. So after birth until adolescence. And the formation where we have some of that hyaline cartilage are the places of where it's left. So one is at the epiphyseal plate, and then the other one here is at the articular. This is the articular cartilage. And so we'll see we also have the formation. This is our spongy bone that's formed here. And so we talked about here that secondary ossification center results in that secretion of that spongy bone here. So spongy bone is in the anterior. There are no medullary cavities that are going to be formed at the epiphysis, and the completion of secondary ossification center results in hyaline cartilage at the locations. One is at the epiphyseal plate. 2 is going to be at the articular cartilages. So before we get into intramembranous ossification, one of the things I wanted to make note of, at the very top here, the perichondrium is going to be replaced with the periosteum, and that is termed ossification. So let's move on to intramembranous ossification. One of the fundamental things with intramembranous ossification is that we are starting off with the fibrous connective tissue membranes. One of the similarities between intramembranous ossification and endochondral ossification is we do get the use of our mesenchymal cells. Another big difference here is that intramembranous ossification, what that is going to help form is the cranial bones, as well as the clavicles. So during endochondral ossification, that is going to be the primary process that's going to be used to build the majority of the bone, and intramembranous ossification, this is specifically for the cranial bones as well as the clavicles. This process here is going to occur about week 8 at the end of month 2. So let's take a look at some of the different steps here for this process. One thing that we've already talked about is the use of mesenchymal cells to get the production. They're going to differentiate into our osteoblasts. We remember that osteoblasts, these are the cells that are going to secrete osteoid, which is the bone tissue. One of the similarities we can also see is the formation of our ossification center. So that occurs here during this process, which is similar to endochondral ossification. Moving onwards to step 2, we have the formation of what we've already talked about here, the formation of osteoid. These osteoblasts, what they will develop into is these osteocytes. So as they're secreting this osteoid, what will happen, they mature and they get stuck here at this spot here. So they get stuck and they become osteocytes, and they're trapped within that part that I just went around that's the lacunae. They get trapped within the lacunae. So this is going to get calcified during step 2. And then moving on to step 3. We get the formation of immature spongy bone, and we also have the periosteum formation. So you can see in this step, we don't necessarily have that periosteal bud, but we are increasing the blood supply here. Make note of, there's no osteoclast going on here, right? If we were to compare this with endochondral ossification, some of the differences, we've already talked about the type of, the tissue is different, what we're starting off with. There's no bone collar formation. Another difference, there is no obviously there's no cartilage here, so there's no cartilage deterioration. And the last thing, number 3, because we're not getting the formation of there's no medullary cavity. Once again, no osteoclasts are going to be involved during this process. So we talked about how this is, for step 3, it's similar in that you get the formation of spongy bone. The fundamental unit for spongy bone, what we term it, is trabeculae. So the last step here is step 4, and we can see that we get compact bone. Compact bone is going to replace some of that immature spongy bone, just deep to the periosteum. And then you also can see the formation of red bone marrow. So red bone marrow is going to start to develop. The process that's going to occur here is hematopoiesis, in which we get the production of our different blood cells.